And this way we can test the equipment before George has to come up and go. I don't know. All right, so George comes to us from great state of, I guess it's Commonwealth, Virginia, right? Got to get that right. So he got his bachelor's degree at Virginia Tech, and he spent a year of that time in New Zealand at the University of Canterbury, and he actually got his BS from Virginia Tech, but then he got this thing called a COP, which is a Certificate of Proficiency from the University of Canterbury, which I'd never seen before, but that was kind of cool. And George thought I was too, because he always includes it in the CV, even though he's not exactly sure what it means. George is a great field biologist. He's got an amazing experience when you read through his CV. The stuff he's done. Oh yeah, so here it is. I thought it was kind of interesting. I think George's journey towards studying birds was kind of set really early on. I looked at the mascots of these places where he went. Right? So Virginia Tech, correct me if I'm wrong, George, but I think it's called the Hokie Bird. Is that right? So the Virginia Tech Hokie Birds, and then he goes to Canterbury, and they actually had a contest, I find out, because they didn't have a mascot. They had a contest. What was it called? It's a falcon. So I think it was kind of preordained from these kind of early educational experiences that he was going to end up working on birds. But he did a lot of stuff. So he spent time in Australia working on pardalotes, which is an amazing little bird, kind of like a little nuthatch. It's a bad analogy, but it's the closest I can come up with. Really cool little birds. He spent some time on the Galapagos Islands studying Nazca boobies. He did camera trapping of jaguars in Belize. He studied red knots. He did bat surveys. He did black-capped vireos in Texas. He's done a heck of a lot of stuff. Okay, so George is a really good field guy, and so. This is George in the field, and I got a chance to go over and work with George. So again, I have to mention that George is co-advised by Eben Paxton. This has been a really special project for me for a lot of reasons, and one of the reasons was it allowed me to collaborate with a former student from many years ago, Eben, who got his PhD here at NAU. And then Eben called me up and said, I got a great prospective student, let's work together on this project, and I jumped at it. And so I got to go over to Hawaii and spend some time with George. And so I got to see what he was like in the field. And the guy, he was just amazing. I mean, he was walking through here, identifying all these birds by songs and calls, pointing out nests, which I admit it, George, half of those nests I never did see. You know, he'd go, it's right there, it's right there. And he'd go, whoa, and after 15 minutes, I finally go, oh yeah, I see it. Can we move on? So anyway, just an amazing field guy. The other thing which that trip allowed me to see was that George is a great teacher, and he was great at outreach. So one of the things that they were doing at Hakalau was to bring these school kids out. Many of them, you know, these are native Hawaiians, and trying to get them connected with the native fauna is kind of tough when most of the fauna is, you know, all the bird fauna they have fauna is extinct where a lot of these kids live. So they have to go to places like this to see it. And George was amazing, and I got some of these photographs, and I just think you can see how comfortable he is leading these kind of groups in outreach. He was also great here at NAU. He TA'd 181 and 182. He did 181 one semester, did 182 3 semesters. He actually whispered to me before we came in here, he goes, I actually really liked 182 a lot more than 181. And I said, that's okay, I understand that. But I looked back at an evaluation I did of George when I went into the 181 lab. So this is the lab he didn't like. And I'll just read a few of things in my evaluation. It says, my overall impression was a class that was used to being run professionally and efficiently. And as I walked out of the room, 2 students ran up to me and said, are you evaluating George? And I said, yeah, I am. I kind of stick out when I come to a class and sit through the whole thing. And I said, yeah, I am. We got to tell you, he is great. He is great. This one woman said, George, he's great. I've had many other TAs, and he is by far the best. He really goes out of his way to provide us material and help. And the other girl's nodding behind him, and she goes, he's better than my lecture instructor. So I thought that was really high praise. I mean, we get unsolicited kinds of comments like that. Plus, I'd seen what he was doing. It just showed what a great educator he was. And then I have to thank George for one other thing. Because he was willing to support, along with Sean, to support the crazy idea of his advisor who wanted to write and put on a program for local school kids about George Washington and the Battle of Trenton. And George, along with Sean, stepped up and we went out to one of the local schools and put this on, and I really appreciated that. But again, it underscores for me how George is willing to go to any extreme to teach, to reach the public on lots of different levels. So George has presented his work at the University of Hawaii, and then just last year in Washington, D.C., he went to the North American Ornithological Congress, presented work there. That sounds like it's a North American meeting, it's actually an international meeting. So he's presented his work in other places. He's got 2 really strong chapters he's going to share with you today. And finally, I just think, this is George in the field. And, a lot of people look at scientists and field biologists like us, and they kind of say, kind of nerdy, But George, he actually pulled it off. I don't know if you saw this cover of GQ, but that was George who rocked it there, right up there. So anyway, I think Eben wanted to say a few words too.
Yeah, but I won't ask you to take your shirt off, George. I just want to really quickly say, so I've had the pleasure of knowing George for 4 years now, working on our project in Hawaii, and I think one of the things that really stood out with George is, he came the first year, we had gotten this money last minute to start looking at productivity, and we were scrambling to get things going, and he jumped right in and did an amazing job. But what he always had was a really strong intellectual curiosity about what was going on. So it wasn't that he just, worked hard and did a great job, but he was always asking why. Why is this way and that way? And then he would just, on his own, go in and read the literature and get all this information. And it was always surprising me. Or we'd be talking about saying, I wonder why that is. He's like, well, I read about this, and someone had seen this, and then, an old study. And I'm like, wow, I didn't know that. So, and I think that really reflects in all the work that he's done. So. Are you looking at that sign? Yeah.
Yeah, it doesn't sound good.
So this one's warm enough. See what happens.
So maybe, the other one?
Why don't you go ahead and start?
Oh, that's what I was wondering.
Hopefully they'll get warm by the time.
Yeah.
You want to try it?
I can.
Go ahead, try it.
It might warm up.
So we'll see. That's great. We warned George this might happen. And it's like it's hard. You're going to have to just do a hula or something.
Take the shirt off.
We got 4 minutes on this one. Why don't you go ahead and start now?
Yeah.
And we'll shift over. Go ahead.
All right. So, obviously I'm George Cummins, and I'm going to be talking about general breeding biology of birds. And I say general just because I'm going to end up talking about 2 kind of different parts as we go through. But first, I'll just say start off with the general theme of my work here. So species are being moved really rapidly across the globe. And this image here just shows human air traffic. So it just shows the connectedness of all the continents where we're rapidly moving, and everywhere we go we're introducing new species. So these new species cause new interactions between the species that are there and those that are recently introduced. So this is one of the big threats, especially when you think of introduced predators into systems. Now, another big change that humans have caused is just overall landscape change. And this can come off of introducing species. This picture here was a forest a couple hundred years ago. But everything you see now is introduced grasses and introduced shrubs since it's been converted to pasture land. And so we see this whole-scale landscape change along with introduced species. But for any species living in those areas to continue to be successful, that starts with its life history behaviors and strategies and how those actually interact with these new environments that they're forced to live in. Now, I specifically look at their reproductive success because that is one of the main parameters for fitness as a species in general, right? And so I focus on some of these behaviors, predator response, and parental care, as those are key components of their reproductive success in general. So my general theme for this talk is going to be, can birds respond rapidly to these novel species interactions and new environments that they're kind of being forced to live in, specifically looking at that reproductive success. Now, as many of you know, of course, I did my work in Hawaii, and I'll get into with each part of my talk why this is a great place to answer some of these questions. But first, I'll just give a broad overview of where I actually did this work. So all my research took place in Hakala Forest National Wildlife Refuge. And this refuge is located in pink on this map. It's on the big island of Hawaii, on the windward or east slopes of Mauna Kea on that island. So it's a very wet place consisting largely of this rainforest that we see here. So again, I'm going to go through two very distinct parts to this talk. So part one is going to be about the predator response of these birds, where I did an experiment to test their responses towards introduced predators as well as historical predators. And then second, I'll be talking about the breeding biology of these birds in a restored forest and comparing how they're breeding in a restored area to that of an old growth site. So first, obviously, part one. Now, why is their predator response important? Well, one of the most important factors in reproductive success of songbirds is nest predation on their nests. If you're going to have success reproducing, you don't want your eggs to end up like this and be broken in the nest. So you should evolve or have behaviors that are trained towards these predators in response to them so that you can have nesting success. However, as I mentioned, there's a lot of introduced species in the world. And these two here, the mongoose on the left and the black rat on the right, are specifically ones that were in Hawaii. So, let's see if this is going to go.
Oh, all right.
I'll move over here now.
I'm just going to move this over.
Yeah, so these two are introduced predators that have been brought to islands, and they're just one of the many examples of predators that we see. And specifically, when talking about islands, a lot of birds have gone extinct due to these introductions. And these two are nest predators. So we see there's some important reasons to be looking at that, but there's no hope. So this bird here is a South Island robin from New Zealand, and this species has been shown to actually respond to introduced predators like rats that have only been there for a few hundred years. So we see some birds that are just going extinct, and then others that are actually responding to introduced predators. So it's worthwhile to figure out what's going on and to try to better understand this system. Now, I'll explain here why Hawaii is actually a great place to look at this. And I'll go through both sort of the history of Hawaii and their responses. Birds may be responses to predators through time. So Hawaii, as most of you probably know, is a volcanic island, or volcanic archipelago rather, in the middle of the ocean that was never connected to a continent landmass. So that means that to get there, you pretty much had to fly or swim. So all these land-based species, for a while, are constricted to the mainland. And that includes things like mammals and reptiles and amphibians. However, birds could get to the islands. Now first, they came from the mainland. And this diagram on the bottom just shows time on the X and general response to a predator on the Y. So that green line high up means that they do have a response to these predators, right? They evolved with them on the mainland. But these birds, along with their predators, colonized the Hawaiian Islands. When they did so, they left behind some key mammalian and reptilian predators that couldn't get to the islands. And so we might expect them to actually lose their response to these predators because they don't have selection pressure anymore. Now, on the other hand, we might think that they could retain response to these predators for millions of years with no selection pressure. But as we've seen so many birds go extinct after the introduction of species, this seems pretty unlikely. Now, the other thing to note about this graph is that probably they're not going to lose their responses to these predators immediately and wholesale. Instead, this shaded area here represents some variation. anytime there's relaxation in selection pressures towards a trait, in this case their response, you might expect that behavior or that trait to become more variable, much like vestigial traits that you hear about even in humans. So under relaxed selection, you'll expect some more variation to appear in that response behavior that slowly then disappears over time. Now, humans, of course, did find Hawaiian Islands, or the Hawaiian Islands, and we brought some predators with us. So we brought some of those mammalian predators that only existed on the mainland to the islands. And this dashed line here represents one scenario that the endemic birds had. Maybe they didn't get any response at all and just flatlined it into extinction, right? On the other hand, maybe the response to these predators is starting to show up in these birds, as we've seen in some of the New Zealand species that are facing some similar threats. But not only did humans bring predators to the islands, we also brought other birds, and these birds still evolved on the mainland. So their response to all predators is pretty high. They evolved on the mainland with those predators. Now, when we brought them and introduced them to Hawaii in the last century, in the 1920s and 30s, they're still living, of course, whoops, that was the wrong button. They're still living, of course, with rats and birds of prey, but the notable exception here is these snakes. Snakes are still not present on Hawaii. So the birds that we've introduced recently are now living there in the absence of one of their main predators on the mainland. And so again, we can expect one of two things. Either they might retain this response over time, shown by this dotted line, or in the past 80 or 90 years, they may have lost this response, or at least it's become more variable through time. So why might we actually expect them to retain these behaviors, even though they're living without one of these predators? Well, there's a hypothesis that's been put forth by this guy, Daniel Bloomstein, and his idea is that because anti-predator behaviors are really similar, that they should be linked to the genes. And what I mean by similar is, if you are going to respond to a predator, first, you're going to have to recognize that that is a predator, and then you're going to have some physiological response towards that predator, some fear response. And after that, you will act upon it. So at the base, any response to predators should be really similar and should be linked, because you don't want to inherit the behaviors towards one predator and not another. That would put you at a selective disadvantage. Instead, you should be inheriting them all as kind of a package. Now, because of that, as long as a particular behavior isn't very costly, especially in terms of your reproduction, you actually should maintain it as long as you're experiencing some predation pressure in the system, even if you've lost some of your predators. So how this might work in Hawaii is, we've got this bird up here, a red-billed beatrix, which was introduced to the Hawaiian Islands by humans in the 1920s and 30s. But it evolved with a full suite of predators represented here, rats and birds and snakes. Now, in the Hawaiian Islands, of course, there are no snakes, right? According to this multi-predator hypothesis, because these behaviors should be linked here, it's still going to retain the response to that absent predator, even over 90 years, which is maybe 30 generations or more for this small songbird. So that leads to my questions here, which, first of all, I want to know, do these endemic birds actually respond to the introduced predators that have been brought to the islands? And then I want to know about these introduced birds and if they're actually responding still to snakes, a historical predator that is not in Hawaii, so they haven't been living with it for a while. Now, I used several species, and I'll show real quick here. First is a group of endemic birds on the left. So I used these four, and just so you remember, they've been living in Hawaii for millions of years now, with only birds as predators, until humans came along and introduced rats and other mammal predators. The introduced species, I'm looking at these two, and they were, of course, introduced last century, less than 100 years ago, and since that time have been living without the pressure of snakes. So, I tested this by presenting models of predators at the nests of these birds, and I used four models. First, the rat model, right? This is that introduced predator that has been brought to the island, so we'll see if the endemics respond to it. Next, I used a snake model. And the snake, of course, represents a predator that's currently not in Hawaii. Third, I had a box. And this box is just a control for a novel object. So we would expect to see the responses higher towards the predators that should be perceived as a threat than to that novel object. And lastly, I used just the branch. And this was just a baseline control of my experimental methods, which I'll show in a minute why that's kind of important in this situation. So these birds nest really high up in trees. So you can't just walk up to most of them. Instead, I used a really long pole, seen here, that extended up to almost 12 meters high. And you can see at the top there was a stick, and I had one of my predator models, in this case a snake, mounted on that stick. And so I did this experiment during the nestling stage. That means that the adults are feeding their nestlings, and then they leave the area to go gather more food. So I would watch when they left the area, I would run over to the nest, extend this pole up, lean it in the vegetation so that it was about a meter or less away from the nest, and looking right at it. Then I would run away and hide and start a video camera and a voice recorder to record all the behaviors. Once they came back, I would record everything for 5 minutes, and then I would just end the trial and go and take the model down. Now, what I recorded was several response variables towards these models. So birds have a response called a mobbing response. And you may have seen this if you've seen small birds chasing a hawk in your yard or something. And this just means that when they see predators, they end up moving around a lot and focusing often that harassment around that predator. Now, humans, when we get angry, we start yelling, and when these birds see a predator, they also make a lot of alarm calls. And so we expect if a bird sees a predator that it's going to have an elevated number of movements and call notes, as seen here when they're really agitated. Now the third thing I looked at was whether or not they fed their nestlings. And that's just sort of a measure of how concerned they are with the situation. So I say that because if you're going to feed your chicks and you see a predator at your nest, instead of continuing on to your nest and feeding your nestlings and showing the predator exactly where they are, you're going to instead eat the food you're carrying and start into anti-predator behaviors, as long as you're recognizing that as a threat. So that's why I recorded whether or not they went ahead and fed their nestlings. Now, I took all these variables and I collapsed them into an agitation score. And this score totaled a 0 or a 3 as the maximum. So how I got this was for the number of movements and calls, I looked at that baseline response to the stick, and if they, the number of movements in a particular trial was more than 2 standard deviations above that number towards the baseline, I gave it a 1. If not, it was a score of 0, showing not quite as elevated of a response. So when you, and then for the whether they fed their nestlings, of course, if they did, that was a 0, and if they did not feed their nestlings, that was a 1. So that gives you this total score of 3 for a maximum level of agitation and 0 for the minimum here. Now, I'll show a video here, and this video is an example of the box trial with the Leothrix, and this is an introduced bird. And this video is just an example of a low agitation score trial. So you won't be able to see movements too well, I'm sure, but use your ears. That's where the nest is in the model itself. And what you want to listen for is any little short notes. Like that one. So this bird is actually going into the nest to feed its nestlings, and it ends up, it's in there right now, feeding those nestlings, right in the middle of the screen. And eventually you'll see a little red line, which shows when the bird comes out of the nest and begins looking at the box. So right now it's inspecting that model, and you don't hear a whole lot of noises. But eventually the bird does leave the area, makes a few more chatters, and then I can say, before this trial was over, they went ahead and came back and fed at this nest a second time. Now this is the exact same nest with the same birds, only a day apart. And this is the snake. So just a different model is the only difference here. Again, the nest is right here in the middle, and the model is already there, but you may not be able to see movements. Instead, just listen out for those harsh chatter notes, and you will know when they saw the predator or the model there. So there's a lot of chatter noises going on. And what you may not be able to see is the movements that are also taking place. So these birds continue to do this for the entire length of the trial until I actually go up there and remove the model. And the whole time, they end up swarming around this area and bouncing and forth between the vegetation, just focusing right there on that model, especially the front end of it. So, what is my predictions for my results in sort of a data form? Well, I use the agitation score here on the Y, and for these endemic birds, I predict that blue bar represents their response to the rat. And I predicted that they would actually have a pretty high agitation score towards the rat, that introduced predator, but not, of course, towards the snake, something they haven't seen for millions of years. Now, like in New Zealand, they do show some response toward these rats, so I thought the same thing might be going here, since these birds are still around in the presence of rats. Now, for the introduced birds, I of course thought they would respond to the rat, but I also thought that they would respond to the snake. According to that multi-predator hypothesis, they should be retaining these responses at least over a time span of 90 years. Now there's another side to this, not just that average agitation score, but also how variable is it between individuals. So this is just a prediction here for kind of an ordination. And really what you need to get is that the dots that are really close together mean that those trials had really similar responses by those birds. And each individual dot represents one trial. So if the dots are split apart, that means that those trials had different responses. So you'll notice for these endemic birds here, I predict that they're going to have a tight clustering of blue dots. So just a tight cluster in their response towards the rat, meaning that it should all be the same between individuals. Now in contrast, their response to the control models in that snake that they haven't seen for millions of years, I expect that to be much more variable. So we say they either lose their response, when things lose their selective pressures on a trait, it'll become more variable. And if you've never experienced something before, like say a box, there's no particular way that you should be responding to that has evolved. Now, for the introduced species, I do expect the snake and the rat to have a tight, similar response between all individuals, showing again that they've retained that response towards snake, but those control models should show a lot more variation. So what did I find? Well, we'll start with these endemic species right here. So again, the agitation score is on the Y, and this has a maximum score of 3, right, and a minimum of 0. And the sample sizes are indicated by these numbers at the tops of the bars. And above those, you'll see letters, and those denote the statistical significance. So letters that are the same means that they're not really different. Letters that are different above bars mean they are. So, for example, here with these endemic birds, we see they have the highest average towards that rat model, but it's only significantly different from that stick. It's the same as that box, that novel object. So we're not getting a strong statistical difference in their response towards these introduced predators. Now when we look at the introduced species, we see a pretty similar pattern. We see that this rat, again, the response to the rat is the highest in its average agitation score, but it's not significantly different from the box. However, we see that it is not what we predicted. So the snake, which is in yellow here, we predicted would be just about as high as the rat was. And in this case, we're actually seeing the rat is significantly higher than the snake. So that would indicate they're not responding to this predator anymore. But if you remember, that video I showed was one of those introduced birds in response to the snake model. So I thought maybe there is something going on there. And I looked at these by individual species. For the endemics, I'm not going to show their individual species graphs because the pattern was pretty much exactly the same, in that they all had the highest agitation score average towards that rat, but it wasn't significantly different than their box trials. However, I did find a big difference between these two introduced birds, the Japanese white-eye and the red-billed leothrix. When we look at the Japanese white-eye, we see that same pattern, really a higher agitation score towards the rat, so they are responding to it, and it's significantly higher than it is towards the snake, which isn't what we predicted. Now, the red-billed leothrix, on the other hand, we see that they are responding to the snake statistically the same as they are towards that rat in the average score. Now, again, it's not statistically different here from that box trial, but we are seeing a similar response between the rat and the snake, which we aren't in the white-eye. So that is kind of interesting. So what about that variation? Do we see differences in how variable these individuals are, not just those average agitation scores? So again, these are some ordination plots here, and they just show anytime the dots are close together means that the responses were really similar in those trials, and anytime the dots are farther apart means that they had more different responses. So when we look at the rat, which is the blue dots here, we see for the red-billed leothrix at the top, that really tight cluster, which if you remember is what I predicted, that they should have a pretty evolved tight response towards that predator. However, the Japanese white-eye is a lot more spread out than that leothrix is, which isn't what we really predicted. For these endemic birds, we see that a lot of the blue dots are clustered right here, but there is some other variation across the way, instead of them all being as tight like the leothrix was. Now when we look at the snake, that's the yellow dots here. Again, for the leothrix, they're all pretty clustered here with just a couple dots that aren't right on top of the rat. So that's pretty close to what we predicted, that these guys would be responding towards these snakes. But again, the Japanese white-eye has a lot of variation in where its responses are. And the endemic birds down here, which haven't seen snakes for a long time, have a lot of variation in their response there, just as much as they do towards those control models, which are in green and black in these trials. Now, I know from being in the field that some of these endemic birds actually do respond towards these introduced predators, but it wasn't really showing up for every individual there. So we saw some of that variation, and we didn't see that high difference in their agitation score averages. But I'll give you an example of why I know that some of these birds are responding. So here is an omao. This is an endemic species. It's been living in Hawaii for a few million years. And when presented with the rat, I had some of these birds that actually just went up to their nest and fed their nestlings, even though the rat model was present. Now in neighboring territories, maybe 100 meters away or less, I had the pair of omao physically attacking the rat. So spreading their wings out, puffing their chest up, and just diving down and slamming it with their beak, right? So that's a pretty strong anti-predator response. And I'll say that those individuals that attacked the rat, they did not attack the box, right?